Hey everyone, I'm Dr. Erica Malhart, and today we're going to be talking about the special senses. So the special senses, right? We've got taste, smell, sight, hearing, and balance. So we're going to talk about all these guys today. First, we're going to talk about the chemical senses, which we consider taste and smell, right? We dissolve those chemicals in mucus, in saliva, and then we're picking up on those chemicals as the sense. And then we're going to dive a little deeper into sight in the eye, hearing and balance in the ear. And then at the end, we'll cover a little bit about our disorders and so even though we're talking about just these special senses, right, taste, smell, sight, hearing, and balance, we've talked already a bit about touch, right, all those receptors in the skin, the mechanoreceptors, all those temperature, pain receptors, all those guys also living in the skin, and they are considered a general sense, right? So today we're just talking about those special senses. So the thing about special senses is they have these special sensory receptors. So remember we talked about the receptors in the skin, right? Those mechanoreceptors that are picking up the pressure and things like that. So these guys are going to be a little bit different. We do have some chemoreceptors. Remember those chemical receptors that we've already talked about. But all of these guys, these special sensory receptors are going to be really confined to the head region. You think about where your special senses are, right, in your head. And these receptors are really receptor cells. So I know we've been talking mostly about receptors as being, part of a neuron, but these guys are really neuron-like cells. So they're either these neuron-like cells or they're very small peripheral neurons. So, and we'll talk about kind of which ones are which. And essentially they're picking up this information and sending it to the brain. So let's first talk about our chemical senses. So this is taste and smell, right? So we talked about these being our visceral special senses because they're inside our head, right? We taste in our mouth and we smell inside our nose. And the special words for these guys, taste being gustation and smell being olfaction, okay? And again, these guys are chemoreceptors, so they're picking up on the chemicals in those areas, right? So your food is going to dissolve in the saliva, and those chemicals from the food are what are going to be picked up by those receptors, as well as any sort of airborne chemicals that are going to dissolve in that nasal mucosa, and then the cells pick up those chemicals in the mucosa of the lining of your nose. So let's dive a little deeper into taste or gustation, gustatory. So taste receptors really are found in the taste buds, right? So, but what are these taste buds? Where are these taste buds? They're on the tongue surface, right? Some are on the epiglottis, which we talked about kind of being in the back of the throat, and we'll talk more about the epiglottis when we talk about the pharynx, the back of the throat. But just think really taste buds on the tongue, right? And so we have these different type of papillae on the tongue. They're these little raised bumps, when you stick your tongue out in the mirror and it looks kind of fuzzy. You've got those little bumps all over it. Well, those are all papillae, and some have taste buds and some don't. So the two types with the taste buds are the fungiform papillae, which are these little raised bumps, kind of look like little dots all over your tongue. And then in the kind of towards the back of your throat, we've got the valate papillae. So they look a little bit bigger. They kind of look like a target. So they're a little bit different. Most of our taste buds are on these fungiform papillae. Okay, so let's take a look at those taste buds up close. So you've got a bunch of these epithelial cells all over your tongue, right? So we've got two major types of cells in these taste buds. So if you take a look over here, we're blowing up one of those kind of target looking taste buds, those valate papillae. So we're looking right here in the middle and we're blowing up just this area. And we're even looking at just one little singular taste bud, even though there's multiple lined up on the side of that papillae. So then we get to this picture. So we've blown up just that one taste bud, okay? And then we're looking at just these cells inside of that taste bud. And we have two major types of cells. We have the gustatory epithelial cells. So we said again, some of these receptors are cells, right? So they're not actually neurons, but they're connected to those nerve fibers, okay? So we have that cranial nerve coming in, and we talked about the different cranial nerves that are innervating those taste buds, and we'll talk about that in a minute. But these guys are what are going to be communicating with these gustatory cells. So these are these kind of purplish blue, I guess the other guys are blue, the purplish white looking cells here. So they are what are actually going to be picking up on that chemical that's going to be dissolved and moving into that taste bud pore, that little pore where all that saliva is coming through. Okay, and then at the very base of the taste bud, we've got these blue, bright blue cells, and these are the basal epithelial cells. And essentially, they're there to replace any of these gustatory cells if they get damaged. You know, if you burn your tongue on something hot, right, or you scrape it really hard with something, then you're able to actually replace those cells with those basal epithelial cells. And these gustatory cells have these long little, they call them hairs, but essentially they're microvilli, and they extend out into this pore to pick up all the dissolved chemicals out on the tongue or in the mouth. And this is just a real picture of a taste bud, so you can actually see those cells and what they look like in that little pore there, which is kind of cool. So even if it gets damaged or not, essentially your cells in your tongue, these taste buds are replaced every week about. So if you just, you know, they've got some wear and tear, you're eating some rough foods, some hot foods, so they just replace them every week or so. So we talked about epithelial cells as having a high rate of turnover anyways, so these just kind of follow that rule. So I'm sure everyone has heard of the taste map, right? On your tongue, you're tasting different things. Like some places it's more salty, some places it's more sweet. Well, they've kind of disproven that idea of the taste map, but we do have these five basic taste qualities. So that is true, where we can pick up these distinct different tastes, right? So we do pick up sweet, sour, salty, bitter, and then we do have a new one that they've been talking about is umami. So, you know, you hear about, ooh, it has a lot of umami flavor, right? So pretty much they're picking up on this glutamate taste, or it's kind of salty, a little more than salty. And then they also have found that they pick up water as well. So that's why you are able to kind of taste the different flavors of water. And some people like certain waters over others, right? So, so these are our five basic tastes that we pick up on in water. So how does this information then get to the brain, right? We want to get it to the cerebral cortex, to that specific gustatory cortex where we're picking up taste, right? So how do we get it there? Well, we've got our cranial nerves, and we talked about these three different cranial nerves. Facial nerve being the one picking up the most information from the tongue, right? Pretty much all 2/3 of the tongue. So most of it's going through the cranial nerve, the facial nerve. But we do have the glossopharyngeal nerve and the vagus nerve picking up information as well. So glossopharyngeal is kind of that back 1/3 of the tongue, and the vagus nerve on the epiglottis. So they are picking up all this information from the tongue, and then they're going to send it up to the brain. We make a stop at the medulla oblongata in the brainstem, but don't worry too much about that. The one we want to focus on is the thalamus. So it makes a stop at the relay station, the thalamus, because the thalamus is going to tell it, hey, this is a taste, so we've got to go to the gustatory cortex, and it sends it out to the correct area of the cortex so that we're able to pick up that information and decide what to do with it and what kind of taste you're tasting. All right, so now we're going to talk about smell or olfaction. And we've already been talking about the cranial nerve 1, right? The olfactory nerve. And we talked about the olfactory bulb sitting up here on top of that cribriform plate, sending down those little fibers through the olfactory foramen into that nasal cavity, into that nasal mucosa. So what are we looking at here? So the bulb is up here, right, sitting up on top of that cribriform plate. But then we've got these projections coming down and into this olfactory epithelium. So this olfactory epithelium is just like any other epithelium, right? It's pseudostratified columnar epithelium. And we talked about our pseudostratified ciliated columnar epithelium in the respiratory tract. So all through the nasal cavity, all through down through the trachea, right? We've got our pseudostratified ciliated columnar. So same type of epithelium going on here. But we have 3 main cell types that are happening and doing all the action in this epithelium. So our first type is the olfactory sensory neuron. So these guys are these yellow guys sitting in here, and they are the ones that are actually going to be picking up the dissolved chemicals in the mucus, okay? So through these olfactory cilia, these guys are going to be picking up all those chemicals in the mucus. Our second type of cell is the supporting epithelial cell, and these guys are just sitting there. They are those columnar cells, the epithelial cells, that are just sitting in amongst this epithelium. They're making up the bulk of the epithelium. And then last but not least, we also have a stem cell sitting in here that, just like our taste buds, they're ready to replace those supporting epithelial cells if anything happens to them, or there's just kind of a natural turnover of the epithelial tissue. So remember back when we were talking about the different types of neurons, right? We have our classic neurons with the dendrites and the one axon. Here would be a classic neuron, right? So these guys have lots of dendrites. They've got one axon coming out. So these guys are the classic neurons. And then we had the 2 other types, right? So we had bipolar neurons as well. So we said those guys are really just found in sensory tissue. So here's where we see a bipolar neuron. So we've got our cell body of the bipolar neuron. Now we've got the one dendrite coming out with the cilia, and that's where we're picking up all that chemical information. And then it's going to bring it up through the bipolar neuron, up through the axon, and then it's going to synapse with one of these mitral cells. These mitral cells are definitely more of a classic neuron, right? And they are going to be the ones that are going to have the axon filaments that are going to make up that olfactory tract and eventually that olfactory neuron. Okay. So again, these bipolar neurons, these olfactory sensory neurons, are going to be the ones that are going to be picking up the information via that cilia that's down in the mucus and picking up on the dissolved chemicals, okay? So when we talk about how we make up the olfactory nerve, right? We've got our olfactory tract made up of all those axon. Those axon filaments are what are going to be making up our olfactory neuron. So when we bring in that information to the brain, where does it go, right? We talked about it has to get to the cerebral cortex, but it does not go through the thalamus like most of our other senses. The olfactory sense, the smell, is one of the only ones that does not go through the thalamus. But what it does do is it goes through the limbic system. So it creates these very strong smell memories. If you've ever had that experience, I know for me, if I ever smell geraniums, which is a flower, I always think of my grandma, and it's a very strong sense memory. So it's very interesting how that happens. So there are some disorders that happen with these chemical senses. So both taste and smell, but mostly kind of smell, I think, is a little more prominent in the disorders. So our first disorder, anosmia, is essentially the absence of smell. So it could happen for certain reasons. Probably the most common reason is allergies or colds, right? You get your sinuses are full of fluid instead of air, and it affects the sense of smell. And so that is the most common reason. But you could get, head trauma, injury, certain medical conditions, definitely old age. When people get older, they tend to lose their sense of smell. But what happens with the loss of smell is it also affects the taste, and they're very closely related. We're not going to go into, how they're related and why that happens, but essentially your taste buds can actually only detect a few flavors without smell. So that's why when you have a cold or allergies, your sense of taste is also disrupted as well. Now we do have another type of disorder called ununcinate fits, and these actually distort your sense of smell, which is very interesting. And you can actually have these olfactory hallucinations, or called phantasmia. And essentially, you have these weird smells where you sense, either a foul smell or even a good smell, but the smell is a hallucination. And this can result from a couple different reasons, but usually it's a bad thing, usually after either head trauma or brain surgery, brain tumors, strokes, any of these bad things that can happen and can cause these weird smell hallucinations. Very strange. If you want to look it up, it's very weird. Okay, so now we're going to talk mostly about the eye and ear for today. So let's start with the eye and vision. And in humans, it's really actually very interesting. We rely very heavily on sight as a species. A lot of other species rely on sense of smell, things like that. So it is very different in humans that we rely so heavily on eyesight. And this is just shown even in our brain. So 70% of all those sensory receptors that we've been talking about, the different kinds that we have, chemoreceptors, mechanoreceptors, all these guys, and 70% of all of those sensory receptors are in the eye. That means they're all these photoreceptors. That's a lot, right? Considering our entire body. So that shows how much is dedicated to sight. And then 40% of that cerebral cortex, where all of that information processing is happening, 40% of the cerebral cortex is involved in processing that visual information. So whatever is coming into the visual cortex and all those multimodal association areas processing all that vision. So that's almost half. So that's crazy. And then when you look at the eye itself, kind of our external features of the eye, you're only really looking at 1/6 of the eye surface. So the globe is quite large, but it sits, back in the socket. in the orbit. So just we're looking only at 1/6 really of the eyeball itself. So when we're talking about these external features of the eye, we call them accessory structures of the eye. So when we look at the eyeball and all these other features outside the eye, we've got our eyebrows, right? So those guys have some coarse hairs on them as well as our eyelids, right? So our eyelids and eyebrows both have these coarse hairs, and those guys are helping to decrease the sunlight coming into the eye, trapping any sort of particles that might want to go into your eye as well. And when we talk about the eyelids, we call them palpebrae, so that's another word for the eyelids. And then the separation between the two palpebrae we call the palpebral fissure. So it's just the space in between the two eyelids. That's all that is. And then where the two eyelids come together at either corner, we call the medial and lateral angles or commissures. So this side would be the medial commissure, this side would be the lateral commissure. And then when we talk about the medial commissure, there's this little pink kind of bulbous area that you notice that comes up, and that is the lacrimal caruncle is what it's called. And essentially it's just this little elevation of tissue, and that's where your tears drain off the surface of your eye, and we'll talk about the lacrimal system in a minute. But when we talk about the eyelids, and I think our next slide is going to help us out with these other two things, these tarsal plates and tarsal glands. And essentially, it's a connective tissue within the eyelid itself to help give some structure to the eyelid. And then the tarsal glands essentially are kind of lined up right along your margin where the hairs come out. And essentially they're just modified sebaceous glands right near those hair follicles. Okay, so on this slide is where we can actually see those tarsal plates and tarsal glands. So if we look, here's our tarsal plate. So essentially it's just this connective tissue structure that lives inside the eyelid, so both of them. And essentially it gives it a sturdiness and it gives it some structure. And then the tarsal glands just kind of come out of those tarsal plates and essentially live at the margin of the eyelid. So kind of where, right behind where the hairs come out of the eyelids, the eyelashes. So if you look in a mirror, you can actually see these little tarsal glands, these sebaceous glands, sitting kind of right at that margin of the eyelashes. And they can get blocked with dirt and dust and things and cause styes, right? Those kind of pink zit looking things that people can get on their eyelids themselves. So now let's talk about the conjunctiva or conjunctiva, however you want to say it. And essentially the conjunctiva is essentially just a mucous membrane. So it sits on the surface of the eye and it's transparent, meaning you can see through it. So you don't really know that it's there because it's transparent. But it can get inflamed and it can cause some trouble. But essentially where it sits on the eye itself or on the surface of the eyelids, we call it a different type of conjunctiva. So if it's along the surface of the eyelid or the palpebrae, we call it palpebral conjunctiva. And then if it's along the surface of the eye, we call it bulbar conjunctiva. And then at the bottom here, where the bulbar conjunctiva meets the palpebral conjunctiva, there's a little curve here and it creates a little sac, and that's the conjunctival sac. So it collects that the lacrimal fluid or the tears, and then the tears can drain out this lacrimal system that we'll talk about, okay? So the whole system of creating lacrimal fluid and draining lacrimal fluid we call the lacrimal apparatus. So the lacrimal gland up here, so it's kind of right above the eye, and that's what's going to be producing the lacrimal fluid, or our tears. And essentially it's going to cross down across the eye, keep the surface of the eye nice and wet, and for protection. But when we have too much fluid on the eyes, then we need a place for it to drain. So right at that commissure, that medial commissure that we talked about, there's that little caruncle, and that's where it drains the fluid into the lacrimal sac. Okay, so down into this lacrimal sac, down into the nasolacrimal duct. And the nasolacrimal duct connects that lacrimal sac to the nasal cavity. So, when you have a cold or you have allergies, and your eyes are running, so is your nose, right? So all that excess fluid that you're producing in your eye from that lacrimal gland is draining into your nose, and that's exactly why your nose runs when your eyes are runny as well. So I know in lab we talked about these muscles, these extrinsic muscles of the eye. So these guys are what are going to be controlling eye movement, right? So how you can look left, right, up, down in a circle. So we have 6 muscles that control the eye, and they all come and insert in this, they originate at this common tendinous ring back here. So essentially there's a tendon that wraps around all of these muscles, and that's where they originate. And then they're inserting on the surface of the eye itself. So we talked about that outer layer of the eye in lab, and we'll talk about it again in a little bit, the sclera, the white part of the eye. And the sclera is a very thick layer of the eye, and that is where these muscles attach onto the eyeball itself. So we have a couple of muscles that kind of look and attach at like a clock face. So the lateral and medial rectus are going to be on the left and the right sides of the eye. So like 9 o'clock, 3 o'clock on a clock face. And I have a slide that shows that as well. And then we have a superior rectus and an inferior rectus, and they are going to be what you can do, move your eye up and down, right? And then we have these 2 oblique muscles, the superior oblique and the inferior oblique muscle, and these guys are what are going to be the rotating muscles of the eye. Okay, so when we look at these muscles on the eye like a clock face, we were kind of talking about, this being 9 o'clock, 12 o'clock, 3 o'clock, and 6 o'clock. So it makes sense for how these guys attach kind of the 4 major muscles of the eye here. So we have our lateral rectus, medial rectus, right? So these guys are doing those left and right movements just depending on which eye you're talking about. Then you have your superior rectus and inferior rectus, and they're doing the up and down movements, right? But I think the confusing ones are our oblique muscles, okay? So superior oblique and inferior oblique. So these guys are doing the rotational movements. So if we look at how they attach to the eye or to that sclera, we see the superior rectus kind of comes in right at a oblique angle kind of across the eye. And if we think about how it's going to move the eyeball, if it contracts, right, it's going to be moving this way, and it's going to rotate the eye if we follow that arrow around like this, okay? And then it's going to kind of come out and do this down and out movement, okay? So if you look at how it's going to rotate the eye this direction, right? So that's that arrow showing for the superior oblique movement. And then if we talk about the inferior oblique, here's this muscle down here coming in and attaching kind of at an 7 or 8 o'clock area around this region. Okay, so it's going to attach down here in an oblique angle as well. And if you think it's going to pull the eye this way though, right? So it's going to make the opposite movement and rotational movement and do this up and in movement, okay? So we follow this arrow for the inferior oblique. So it rotates the eye kind of upward and inward, okay? So when we talk about the eye muscles, we talked about those three cranial nerves that innervate them, right? Our cranial nerve 3, 4, and 6, okay? And I said we don't need to specifically know the exact muscles that they innervate, but I want you to know which three muscles and their names and the ones that are doing these movements of the eye. And this cranial nerve 3, or the oculomotor nerve, is really the one doing the majority of the movement, innervating the majority of the muscles, right? And then we have our trochlear and abducens 4 and 6 that are just innervating essentially one muscle each, but fairly important for them all to work together. And if you don't have these muscles or these nerves working correctly, like we talked about in the lecture, you get the strabismus and those weird eye movements, right? So now let's talk about the anatomy of the eyeball itself. So we've talked about kind of the outer accessory structures of the eye, the muscles of the eye, and now we're going to talk about the anatomy of the eyeball itself. So the thing to remember about the eyeball is essentially all these structures are going to help support and protect those very important photoreceptors in the back of the eye, those sensory receptors picking up on the light. So essentially the whole structure of the eye is really to help try to gather, focus all this light that's coming into the eye and focus it right onto the back of the eye, where the best area to perceive light and pick up on images is going to be. So that's the whole point of all these structures of the eye, and to also, bring in nutrients and blood supply as well to these structures of the eye. So when we talk about the different layers, we have 3 different layers of the eye, and we call them tunics. Okay, so these 3 different tunics, or these 3 different layers. So the outermost layer we have is the fibrous layer. Okay, so that's this white layer on the outside. And then in the middle, we have a vascular layer, and anything that you hear vascular, you're going to think blood supply, okay? And then we have the innermost layer, which is our sensitive layer that houses all of those photoreceptors. And we'll talk about each of these layers individually. Now, when you also talk about the structure of the eye, you have some cavities. You're creating these cavities by these outer layers, right? So we have these internal cavities that contain fluid, and the fluid is very different in this back chamber from then in this front chamber or cavity. So we'll talk about chambers versus cavities. And the fluid itself we call humor. I know that's kind of a weird word, but humor is what we talk about as the fluid within the eyeball itself. So they have the anterior cavity and the posterior cavity, just depending on what side of the eye you're talking about, anterior versus posterior. So the outermost layer, or the external layer of the eye, is our fibrous layer, right? So when we talk about the fibrous layer of the eye, we can talk about 2 different regions of that layer. And the majority of it is the sclera. So the sclera is about 5/6 of the layer, and it's the white kind of opaque layer. You can't see through it. It's very dense, and it provides an anchor for all of those rectus muscles that are coming in and attaching to the globe that we looked at earlier. Now, the front 1/6 of the eye is called the cornea, and the cornea is made out of the same material. It's still a fibrous layer, but you can see through it, so it is clear, and that's how we're able to see through the eye and see the iris and the pupil and those structures that are deeper to the cornea. Now we have a couple other structures with this external layer that we need to talk about. And the first one is the limbus. And the limbus is the junction between the sclera and the cornea. So it's just where it changes between the scleral tissue and the corneal tissue, okay? And then at the bottom here, I mean a little bit at the top as well, but mostly at the bottom due to gravity, we have this scleral venous sinus, which allows that fluid in the eyeball that we talked about, that humor, to drain out this scleral venous sinus. Now, we've talked about another venous sinus in the body, and hopefully you remember our dural venous sinus in the brain, which drains that cerebral spinal fluid back to our cardiovascular system. Well, this does the exact same thing, but with the humor or the fluid inside of the eye, okay? So now let's talk about the second layer or the middle layer of the eye, and this is our vascular layer. So it's made up of the choroid, which we'll talk about first, and then it's also made up of the ciliary body, which is right here, as well as the iris, okay? So first we'll talk about the choroid, and the choroid is the majority of the vascular layer of the eye. So it's taking up, almost pretty much 5/6 of the eye, just like the sclera. And this is very vascular, meaning it has a lot of blood supply, and it also has a very dark pigmented color. It's almost like a brown color. And essentially it's made up of a lot of melanocytes. So if we remember back to our skin and what makes up the pigment in our skin, it's melanin, which is produced by these melanocytes. So essentially these guys live within this layer, and what are they doing? They are trying to prevent scattering of light. When the light comes into the eye, we're trying to focus it all the way to the back of the eye. So if any light rays kind of scatter around the globe, these melanocytes and this melanin pigment is going to try to help prevent it from scattering and focus all that light right into our sweet spot in the back of the eye, okay? Now, this picture just kind of shows you how well vascularized the choroid layer is. So you can see all of these blood vessels coming in, and of course you don't need to know all of the blood vessels, but I just thought it was a cool picture to show this choroid or vascular tunic. So the other part of the vascular layer is the ciliary body. So the ciliary body is a thick ring of tissue, so it circles all the way around the opening of the eye. And essentially what happens is it has these little suspensory ligaments that are coming down from the ciliary body that help hold the lens in place. Okay, so the lens is right behind the opening of the eye, and it's going to help bend the light as it comes through into the back of the eye. Okay, but the ciliary body, the whole point of the ciliary body is to have and suspend the lens in place. Okay. The ciliary process is essentially just part of the ciliary body, and it's just projecting back, and it looks a little like a serrated edge, and it just essentially is creating this junction between the ciliary body and this innermost layer of the eye, which we'll talk about next. Okay. So the whole point of the ciliary body, which is still part of this vascular layer, is to suspend and hold the lens in place. Okay. So now the last part of the vascular layer is the iris. So the iris is the part of the eye that we're able to see when we look through the cornea. We're able to see the color of the iris, right? It makes the different colors of people's eyes. It can be blue, green, brown, right? Different colors of those pigments in the iris. So essentially all the iris is is it's a layer of two smooth muscles, okay? So we have two different smooth muscles that make up the iris, and the whole job of the iris is to either constrict the pupil or dilate the pupil. And the pupil is essentially just an opening. There's nothing that's blocking the pupil. Essentially, the lens sits right behind the pupil, but we should be able to see through the lens. So essentially, you're just looking straight into the back of the eye. And a lot of people don't realize that that's what the pupil is. They think it's actually just a black structure, but it's not. It's a hole. And you're just looking into the back of the eye, which is dark. So it just creates this black appearance, but it's just a hole. So, and the hole is created by the iris. So the iris has these two muscles, these two smooth muscles. And one is dedicated to constricting the pupil, and that's the sphincter pupillae muscle. Okay, so that's gonna, think of the sphincter as closing, right? So the sphincter is gonna constrict it. So it's this inner circular layer of muscle, and essentially when it contracts, it's going to create a smaller hole or constrict the pupil. And then we have this outer layer, or the dilator pupillae muscle, and essentially it is this outer kind of radiating type of muscle, okay? So when it constricts or when it contracts, it's going to pull the pupil open, okay? So it's this radiating muscle that essentially is going to dilate the pupil and make it quite a lot larger. Okay, so why do you want to constrict and dilate your pupil? Well, if it's light outside, right, if too much light is coming into your eye, you want to protect your eye and constrict that pupil so you keep the light out of the eye. And then if it's dark, right, and you need some more light into the back of the eye, you're going to dilate the pupil to allow more light coming into the eye. And you can check that your muscles are working correctly in the iris by the pupillary light reflex, or the PLR. And this is part of the cranial nerve test when people are testing that your cranial nerves are working correctly and your eye is working correctly, and they shine a bright light into your eye. And essentially it's to make sure that it constricts correctly and then it dilates back when it goes back into dark. And that's how you can tell that all those muscles are working correctly, and the innervation of those muscles as well. And we talked about those cranial nerves in lecture the other day, causing these muscles to constrict and dilate. All right, so now let's talk about the innermost layer of the eye, which we call the retina. We've talked about the outer fibrous layer, the sclera, the middle vascular layer, the choroid, and now we're going to talk about the neural layer because it has the neural tissue that's going to pick up on that signal and send it to the brain. So if we take a picture here, kind of take a chunk out of this bigger picture and blow it up, and we look at this lower picture here. So now we can actually look at kind of the layers of the retina, because we have two layers. So we have this outer layer, or this pigmented layer, and essentially it's just a single cell layer of melanocytes. And the melanocytes, just like we talked about in the choroid, are making that pigment, that melanin, to create kind of a dark layer to help the scattering of the light in the back of the eye. Then the rest of the retina is made up of this neural layer, or a sheet of nervous tissue. And we have three main types of neurons, or even these cells. So some of them, remember we talked about kind of these specialized cells, and these neurons are what make up these sensory receptors, okay? So first we have our photoreceptors, right? These guys are the main players picking up on that light in the back of the eye, and we've got two different kinds. We've got cones and we've got rods, and we'll talk about the difference between the two in a second. So connecting those photoreceptor cells, we have bipolar cells, and these guys are bipolar neurons. Remember, we talked about the cell body being in the middle, then we've got some dendrites and some axons on one side and the other. And these guys are going to connect up to these ganglion cells. And the ganglion cells are important because their axons are going to all come together and they are going to form the optic nerve. So essentially, if we go back to our bigger picture, we have our retinal layer here, and then we have all those axons from those ganglion cells all coming together, and they're all going to form a bundle of a nerve, and that creates that optic nerve, which is sending all that information back to the brain. So now we have an even more blown up picture, and just I've written out for you what is happening in the back of the eye. So essentially the light comes in, right? So the light's coming in and it hits the back of the eye. And that's why we have these pigmented cells in the back of the eye. They're going to help from scattering light, and they're going to focus all that light into these photoreceptor cells, okay? And they are going to that's what's picking up on that light. And they are going to transmit that signal to these bipolar cells, which are then going to generate a nerve impulse and signal these ganglion cells. And then again, those ganglion cells are really important because they're the ones that are going to create that optic nerve from all of these axons, okay? So they're going to converge all to create a signal to the brain. So the light comes in, creates a signal, and those photoreceptor cells are then going to pass that signal up into the optic nerve and out of the eye and to the brain, okay? So now when we talk about our two main types of photoreceptor cells, most of us know that we've got rods and cones in the back of the eye, but we're not necessarily sure what that means or what they do. So when we talk about the rods, these guys are really sensitive to light, and so they allow us to actually be able to see in very dim light, so they can pick up on just a very small amount of light. So usually animals have a majority of rods because they're able to see better at night than we are, right? So we have a lot more cones. So cones operate best in bright light, and these guys are really our high intensity vision. So they are able to let us to see very high intensity images, very clear images. And it also allows us to see in color too. A lot of animals can't see color as well as we can. So these guys, even though we call them photoreceptor cells, they are considered neurons. So if we even get closer in, so now we're going to blow up the retina. Okay, so here's our retina. That's that outer pigmented layer, and then we've got our photoreceptor cells, our bipolar cells, and our ganglion cells. So if we just blow up our photoreceptor cells and take a look at them, we have these outer and inner segments, and I don't want you to worry too much about them, but essentially the outer segments are what are really going to be picking up and absorbing the light and absorbing these visual pigments, okay? So these guys are what are going to be picking up the light, and then they're going to talk to these inner segments that are going to actually generate the nerve impulse that talks to that bipolar cell, okay? So the thing about the photoreceptors, though, is they are very, very vulnerable to light or heat. And you might say, well, we need the light, right? That's what they're picking up on, their light absorbing pigments. But they are actually very, very sensitive, and light can actually destroy them. So that's why we have our constricting pupils and our eyelids and our eyelashes and our eyebrows to really help decrease the UV light that is coming into our eye. Because if we destroy them, we cannot regenerate them if they are destroyed. But we can replace them. We can replace the outer segment, and that's part of the job of the inner segment. So the inner segment can replace the outer segment. But if the whole cell dies, that cell body of the photoreceptor dies, we cannot replace them. So we are able to renew and regenerate these outer segments, but if the inner segment or that cell body dies, we can't replace them. So now that we know our different layers of the eye and we've got some cavities full of fluid in the eye, let's talk about some specialized regions of the retina, okay? So when we talk about the retina, we're really just talking about this posterior 5/6 of the eye, okay? These guys have the photoreceptors, they're picking up all the light. But in the front, we have this delineation between the retina and our ciliary body, and we call that region the ora serrata. And we've seen this serrata word before when we were talking about the serratus anterior muscle. Essentially, it's just that serrated edge look. So essentially, it creates this serration between the ciliary body and the retina. Now, we're trying to focus all the light into the back of the eye, right? So all this light is coming in and we're trying to aim it right at this spot in the back of the eye. And that spot is called the macula lutea. And we want it to go back there because that's where we have the majority of our cones. And we said our cones are essentially how we get very good vision, very clear, colored vision. So we want all the light to focus back there. Now in the very center of the macula lutea, we even have an area that has only cones, and that's called the fovea centralis. So the fovea centralis has the highest visual intensity right at the center of the macula. And so that's where we're trying to really focus all of our light. And that's why you can see things the best when you look straight at it versus looking at something per your peripheral vision, right? Now, at the back of the eye, we're trying to focus all this light in the back of the eye, but right below that is the optic disc. And what the optic disc is a blind spot. And why is it blind? Well, we have no photoreceptors there. So this is where all of those axons exit the back of the eye and create that optic nerve. So the optic nerve is exiting the back of the eye, but also you have blood supply coming into the retina as well. So you have a nerve exiting and a blood supply entering. You have no cones or rods in that area, so it is a true blind spot. Now, it is very close to the macula lutea where we're trying to actually focus all the back of the light into the back of the eye, which is quite interesting that it's so close. But it's also why if you were looking at the models and things like that, you notice that the optic nerve is not coming out right at the back of the eye. It's a little inferior. So we don't want it right in the back of the eye because that's where we're focusing all of our light so that we can see the best. So when we're talking about the retina, we're also worried about the blood supply to the retina because we're trying to get the best blood supply and nutrients and oxygenation to those photoreceptor cells. So the retina actually receives blood from two different sources. So when we're talking about the outer kind of 1/3 of the retina, we're talking about capillaries coming from the choroid. So the choroid, remember, is that vascular layer that has a bunch of blood supply already. So it's sharing that blood supply from the choroid to the retina. And then the innermost layer, since it can't quite reach the choroid, it has its own blood supply. So remember we talked about the optic disc, and the optic disc has the optic nerve coming out from the retina, but then we also have some vessels coming in. So we do have a central artery and vein coming into the retina to help supply the retina with oxygen and nutrients. So if you look at this image, this is what the eye doctor is actually looking at in the back of your eye. The whole thing is the retina when you're looking in the back of the eye. And then this bright spot here is the optic disc. So that is where the optic nerve is coming out and the vessels are coming in. So you can trace and look at all those vessels, and that's what the doctor is looking at, the health of these vessels and arteries coming into the back of the eye. And then right near that, to the left here, you have the macula lutea. And so that is that sweet spot that we're trying to focus all the light into the back of the eye. Okay, so since we've talked about the retina, and now we know that we want to get all of this light into the back of the eye for those photoreceptor cells to pick it up, let's talk about some structures that help focus all of this light to the back of the eye. So when we're talking about these internal chambers or cavities of the eye and the fluid that fills them, when we talk about the different chambers, we look at the lens. So we essentially create a line from the lens and those ciliary bodies, and in front of that, or on the anterior side, we have the anterior segment or anterior cavity, however you want to talk about it. And when you talk about the posterior segment, it's behind the lens. And we're going to talk about that posterior segment or posterior cavity first. And we said all the fluid in the eye is called humor, but essentially we have two different types of humor. Haha, you could make a joke right there. So the back of the eye is filled with vitreous humor, and vitreous humor is very jelly-like, so it actually has a jelly-like consistency, kind of like a grape, okay, the like internal guts of a grape. That's kind of what it feels like. So what is the job of this vitreous humor in the back of the eye? What it does is it helps to transmit and focus the light coming into the back of the eye. So it's going to help focus that light, but also it helps to support some of these structures in the eye to give the eye pressure and maintain that intraocular pressure. So it supports the back of the lens, so it helps to put some pressure on the lens, but it also really helps to keep the retina tacked it down to the back of the eye, or attached to the back of the eye, because the retina is really flimsy and it can actually come detached from the choroid. So you've heard of people maybe having a detached retina, and that is a big possibility. But what the vitreous humor does is it tries to help put some pressure and keep the retina attached to the back of the eye. All right, so now let's talk about the anterior cavity or anterior segment. We draw our line through the iris and ciliary body, and that divides posterior and anterior. So now we can further divide this anterior segment into chambers. So we have an anterior chamber and a posterior chamber, okay? So the anterior chamber is between the cornea and the iris. And then the posterior chamber is between the back side of the iris and the lens. Okay, so that posterior chamber is quite a bit smaller. Okay, so when we talk about the anterior chambers or the anterior segment, it's full of aqueous humor. Now, aqueous humor is different from that vitreous humor that we have in that posterior cavity. This aqueous humor is a lot more fluid-like, water-like. It's not that jelly-like consistency like our vitreous humor. So it has a different function and it comes from a different place. So the aqueous humor is continuously produced, and it's produced by the ciliary body. So I kind of lied when I said the ciliary body only has one job to hold that lens in place. It also has a job of producing the aqueous humor. So it is a blood filtrate, just like our cerebrospinal fluid. So you can think of the ciliary body just like that choroid plexus in the brain, in those ventricles, right? Producing that cerebrospinal fluid. So it's just a blood filtrate, meaning the capillaries sitting in here in this ciliary process or ciliary body, and it's just leaking out the fluid from the capillaries, leaving all the blood cells in the vascular tissue in those capillaries. But then all of this fluid is coming out into these chambers, and it's helping to nourish the lens and the cornea, because the lens and the cornea have very poor blood supply. So they're far enough away from the vascular tissue in that choroid so that it just doesn't get very good blood supply. So this is really the only way we can get nutrients to the lens and the cornea. So now let's talk about the lens. It helps delineate between the anterior and posterior cavities of the eye, but its main job is really to focus and bend the light into the back of the eye. We want it to bend it right so it hits that sweet spot in the back of the eye. And essentially it's just a biconcave disc. Okay, so biconcave means it has two curved edges, right? So that's what helps to bend that light. And it's made up of these lens fibers, which essentially are just protein fibers, and they're packed together kind of like an onion. So they're packed in these layers, and they're very precisely folded in a very specific way. And essentially the very precise folding of those proteins makes it transparent. So you should be able to see through the lens. Okay, so when you can't see through the lens, you get cataracts. So a lot of people get cataracts in their eyes, and what happens is it's just a misfolding of those proteins, and then you get these opaque regions where you can't see through it. It's not transparent anymore. And a fun fact about the lens too is there's continuously, you're adding new fibers, new protein fibers to the lens, or new layers. And essentially the lens just enlarges throughout your life. And if you do have problems with the lens, what they do is they can actually just put in a fake lens into that area to help bend the light. So that's what cataract surgery is it takes out those misfolded proteins in the lens, and it takes out the whole lens and replaces it with a fake lens. So now what the whole eye is trying to do is to bring in the light into the back of the eye and bend the light rays so that they converge at a single point on the retina. And that single point we're looking for is that macula lutea and that fovea centralis. So essentially you want to bend the light so that it converges right on the sweet spot. So the structures that are doing this bending, and I want you to know these three structures that are bending the light. And it's the lens. So we just talked about the lens being the big bender of light, but we do have two other structures that help as well. The cornea itself bends light, right? So the cornea is the first thing that the light passes through when it's coming to the back of the eye. Then it hits the lens, and as it passes through the whole internal structures of the eye, it goes through the humors. So we have aqueous humor in the front and vitreous humor in the back. And the vitreous humor in the back is what really helps to bend the light in the back of the eye, okay? So what happens is that we can adjust the bending of the light, okay? And this adjustability of the lens is called accommodation. So we can adjust the curvature of the lens essentially by either flattening it out or bulging it. So our two different pictures here, our lens is a little more flattened in this first picture, and it's a little more bulging in this second picture. And the only thing I want you to know about this is that the innervation of those ciliary bodies is what is causing this difference. So we're talking about the autonomic nervous system, right? You don't really have control over either stretching your lens or bulging your lens. That is done via the autonomic nervous system or the involuntary nervous system. So the only thing I want you to know about that innervation is when you have the sympathetic innervation, that fight or flight response, essentially you're creating a lens that is looking better at distance. Okay, so this is the first picture. So you don't need to associate sympathetic with the more flattened lens, just associate sympathetic activation with distance. Okay, you can see better farther away, right? If you think about it, you're trying to fight or flight, right? Something's running at you. You want to be able to see that distance very, very well, okay? Versus parasympathetic, right? Parasympathetic is just your rest and digest. So you don't need to see the tiger coming across the field. You just need to see your food and what you're going to put in your face. So that is just looking at things that are at close distances. Okay, so that's the bulging, or our second picture here. And again, you don't need to know parasympathetic is bulging, just that parasympathetic is for close vision. Okay. So kind of cool how our eye can accommodate and see further away and close up. So now, how does this visual information get to our brain, right? Our visual pathways to the brain. We've talked about the first part about how our light comes into the back of the eye, which activates those photoreceptor cells, right? And then those photoreceptor cells are responsible for generating that neural signal that's then transferred to the bipolar cells, then the ganglion cells, through those axons and exiting as the optic nerve. Now, where does the optic nerve go? We've talked a little bit about this in lecture as well. So most sensory information has to go through the thalamus, right? So we're going to send that information through the thalamus. And of course, you don't need to know which nucleus of the thalamus, but just through the thalamus. And then essentially back to the visual cortex, right? Our primary visual cortex. And the thing about the eye, and we talked a little bit about this in lecture as well, but now we're going to give it a name, and it's called partial decussation. So we talked about this decussation and this crossover event in the central nervous system and why we control, the opposite sides of our body. Well, we only have a partial crossover in the eyes. So essentially, the information coming from one eye is sent to both sides of the visual cortex. So both hemispheres. So we send some information one way, some information the other way. Same for the other eye. And this gives us our depth perception, right? We kind of tested that in lecture. We covered one eye and you try to touch something and you can't really tell how far away it is, and that's your depth perception. And that's all because you have this partial decussation happening in that optic chiasm of the brain, okay? So we do have some disorders of the eye and vision, and we've talked a little bit about lens cataracts already, and that's our first picture up here. So this is a lens that contains some cataracts, so it's not translucent anymore. You can't see through the lens. So a lot of times you undergo cataract surgery to replace that lens. But more common disorder is just not being able to see far away and not being able to see very close up, so nearsighted and farsighted. So essentially nearsighted, the technical name is myopia, and you don't need to know that, but essentially the eyeball is too long, so that focal point, that sweet spot, is not quite hitting where you want it to hit, okay? And I have a better slide coming up to explain nearsighted and farsighted. And then farsighted, you can't see close up, so the eyeball is too short, and essentially that good sweet spot falls too far away, too behind the retina. So again, you're missing that sweet spot, okay? And then glasses can help adjust and help focus that light right onto the sweet spot. So we'll talk about that. So we talked about cataracts already. And then a strabismus, we kind of talked about that in class as well. So what happens is if the eyeball is deviated one direction or the other. So essentially this is called a medial strabismus in this picture because this left eye is pointing medially, okay? So there's a weakness or a paralysis of one of those eye muscles. And in this case, can you guess maybe which muscle might be affected? Or even a better question, what cranial nerve might be affected? So it could be the cranial nerve affected, or it could be the muscle itself. So hopefully you were kind of thinking maybe it might be this lateral rectus, and the lateral rectus has its own nerve supply via that abducens nerve. So just kind of an interesting thought process there, and not something I would ask you on the exam, but just kind of an interesting problem. So here's that picture to show you the difference between nearsighted and farsighted. So when you're nearsighted, you can't see far away, right? So nearsighted, you can see near. So essentially what happens is the eyeball is too long and that focal point, that sweet spot, misses the back of the eye, right? So you put on a corrective lens so that you can adjust that focal point right to the back of the eye. And opposite for farsighted, you can't see close up, your eyeball is too short, so your sweet spot misses and goes too far or too beyond that sweet spot. So you put a different type of corrective lens to bend the light appropriately so that it'll hit the back of the eye correctly. And again, you don't need to know, the different types of lenses and things like that to be able to adjust these nearsighted and farsighted. All right, last but not least, we're going to cover some of the embryological development of the eye. And this is very complex, but I'm just going to show you what I want you to know, okay? So the eye actually develops from an outpocketing of the diencephalon. So remember our diencephalon, the middle part of the brain, the thalamus, the hypothalamus, the epithalamus. So it's coming from that embryonic diencephalon, okay? So there's two parts that I want you to know, okay? So the overlying ectoderm, so that's the light blue outer covering. Remember, we have our three kind of germ layers, our ectoderm being the outermost layer. And essentially in this one area, it's going to thicken and become the lens placode. And the lens placode becomes a lens vesicle, which will then become the lens, okay? So that's how we create the lens. Now, how we create the retina is actually from the diencephalon itself, and we create this little optic vesicle. The optic vesicle is just a little outpocketing of the diencephalon, and so that optic vesicle creates this optic cup, which then becomes the retina, okay? And you can read a lot more about these embryonic developments in the textbook and things like that, but this is just all I want you to know for this class. And this shows you a little bit more about the progression of those lens vesicles and the retina, okay? So now we're going to spend the rest of this lecture talking about the ear. So the ear is responsible for both hearing and equilibrium, right? So we've already talked about our cranial nerve responsible for hearing and equilibrium or balance, and that's our vestibulocochlear nerve or cranial nerve 8. Right, so within the ear, we've got three major regions that we talk about. And the outer region or the external ear is really responsible for hearing, right? So we'll talk about those different structures in the external ear or the outer ear. And then in the middle ear, we've got some bones in here, or ossicles, that we'll talk about, and they are also responsible for hearing or transmitting the signal into the internal ear or the inner ear. And now the inner ear is both hearing and balance or equilibrium. So that's where we finally find our balance center is in the inner ear. So let's take a look at some of these outer or external structures. So the outer ear, right, what we can see when we look at each other, right, is the ear. But the technical term for that is the auricle or pinna. And really, it's kind of like a funnel. You know, you've seen some species that have better hearing than us, and they have fairly large ears that helps to direct the sound into that external acoustic meatus. So it brings in the sound, funnels the sound in so that the sound will travel into these inner ear structures. Now, this auditory canal or external acoustic meatus has some special things in it. We have some hair, some sebaceous glands, ceruminous glands. These guys are just the ones that make earwax. So there's just some special things about the ear, this auditory canal, and it helps to just trap particles before it enters the inner ear structures. Now, the tympanic membrane is the boundary between our external ear and the middle ear. So a lot of people, call this the eardrum, but the technical term is the tympanic membrane. And essentially, it's very similar to a drum, and essentially it vibrates with the sound coming in from that external acoustic meatus. And then it's going to vibrate these ossicles in the middle ear. Okay, so we'll talk about that in a minute. So the tympanic cavity is another word for this middle ear. And essentially it's just a small space full of air, so it's not full of fluid, it's full of air, and it's inside our temporal bone. So remember the temporal bone, kind of right under the ear. This is where these structures are housed. So we have our tympanic membrane kind of on the outside, and then we've got two holes on the inside. And the first one is the oval window. Okay, so the oval window is covered by one of those ossicles. Okay, the oval window is essentially kind of the entrance into these inner ear structures. And then we also have the round window, so two of these holes, these oval window and round window, and essentially it's just a flap that helps with the equilibrium between the inner ear and the middle ear, and it flaps open. Okay, and I have a video that I have posted on Canvas for you guys to help kind of see some of these structures in action. So the other part of this middle ear is the pharyngotympanic tube, or what we call auditory tube. Another word for it is the eustachian tube, which you'll hear more often used in the medical field. And essentially it connects the middle ear structures to the back of the throat, or the pharynx. So this allows for equalization of pressure. When you go on a flight or you pop your ears, right? That popping sound is essentially opening up this eustachian tube to equalize the pressure between the middle ear and the external environment. That's the whole job. But if you're sick or anything like that, or have a ear infection, a lot of times this tube is blocked, and essentially you can no longer equalize the pressure within your ear, and it causes a lot of pain. So if we look inside that tympanic cavity, we find the smallest bones in the body. So we've talked about all the other bones in the body, and these are actually the smallest bones, and they're called auditory ossicles. So ossicles referring to bone, okay? So our three bones in here, the first one is the malleus. So the malleus is attached to the tympanic membrane, okay? And it's also termed the hammer because it kind of looks like a hammer if you look at it. And then it's attached to the incus. Okay, the incus would also we call the anvil, because at a certain angle, if you look at it, it looks like an anvil, but nobody really knows what an anvil looks like anymore. And then the last ossicle is the stapes, which this one actually does look like a stirrup. So if you turn it over sideways, it looks like a stirrup that you'd put your foot in a saddle. And so what happens is this tympanic membrane vibrates with the sound waves, and essentially it pushes on these ossicles, and essentially it's going to eventually push on that oval window, and then that's going to vibrate all of these internal ear structures, okay, and deliver that sound wave into the inner ear, okay. So now what are the internal ear structures? And we term these internal ear structures as the labyrinth. It looks kind of like a maze of some circles and kind of some snail shell looking stuff. And so we term all of these structures together as the labyrinth. And these guys also live within that temporal bone, just like our middle ear. But the bony labyrinth consists of three main parts. So these are bony structures that are part of that temporal bone. So the first one is the cochlea, and essentially it looks kind of like a snail shell, right? So it's kind of circular, and that is what houses our hearing structures. So we'll talk about that. And then we have these two other structures on the other side. One is kind of a rounded structure called the vestibule. And then we have these circles called the semicircular canals. So the semicircular canals and the vestibule, these two structures together house the structures for our balance and equilibrium. Okay, and you can even see how the nerve splits here, and the cochlear nerve comes into the cochlea, and then the vestibular nerve comes into the vestibule, okay, with connection to those semicircular canals. Now, when we're talking about the internal structures, we have that outer bony labyrinth that is part of the temporal bone itself. But then inside that bony labyrinth, we have a membranous labyrinth. And essentially, these guys are membrane-walled structures or sacs, ducts, as you will, and these guys are full of fluid. So these sacs full of fluid are living within the bony labyrinth, okay? So they are associated with the bony labyrinth that house these membranous structures. So there's three main parts, and they coincide with our bony labyrinth. So if we look, we started with a cochlea and the bony labyrinth. So we have our cochlear duct that is a membranous sac full of fluid, and it is inside the cochlea, okay? So that is our hearing part. And then if we go over and look at our vestibule, we actually split it into two different sections, okay? We have the utricle and the saccule. So both of these rounded structures, so here's the saccule and here's the utricle. So you can see the utricle kind of attaches to those semicircular canals, whereas the saccule kind of somewhat attaches a little bit to the cochlea, Okay, but both of these structures are housed within the bigger bony vestibule. Okay. And then last but not least, and kind of the easiest, are the semicircular ducts, because they just live right inside of those semicircular canals. So when you say semicircular canal, you're referring to the bony part. When you talk about semicircular ducts, you're talking about the inside fluid part of those canals. Okay. So let's start with the cochlea first. So this is this spiraling kind of snail-like structure inside that bony labyrinth. And essentially it creates this pillar of bone. So all these tubes kind of spiral around, right? So if you continue that spiral, it looks like this. And then in the center you get this bony-like structure. And this bony structure is called the modiolus. And that is what houses the cochlear nerve fibers. So that cochlear branch of the vestibulocochlear nerve is going to come in to this modiolus and send off these little branches to each of these spirals within the cochlea. So the cochlear duct is that membranous labyrinth which houses the fluid, but it also contains a very special structure called the spiral organ. So the spiral organ, and we blow up just one of these spirals over here to get this picture, and then here's our spiral organ right in the center, okay? So it is connected to our cochlear nerve fiber. So this is where we have the sensory innervation. Okay, so we have this receptor epithelium, just like we've talked about with our smell and taste, right? We have these receptor epithelial cells that essentially are going to be transmitting the signal to the cranial nerve, which then goes to the brain. So these special receptor epithelial cells sit on this basilar membrane, and essentially the basilar membrane is what is going to transmit that signal. Okay. Now these receptor epithelial cells, we call them hair cells. So we have these little hair cells that sit on the basilar membrane, and they are what are going to be picking up these vibrations in the sound and sending that signal up through the cochlear nerve to the brain. Okay, so let's and look at this spiral organ with these hair cells a little closer. Okay, so we've blown up even our little triangle, this spiral organ, okay, and then we've taken that and now we get this blown up picture. So here is that basilar membrane on the bottom, okay, and then we have our nerve fibers running from the cochlear nerve up into these epithelial hair cells. So they have these little hair or stereocilia sitting up at the top of these cells, okay, so that's why they call them hair cells. And we can actually divide the hair cells into the inner hair cells, which are a little closer inside, and then the outer hair cells. And they have two very different jobs. The inner hair cells are what are actually transmitting the vibrations to the basilar membrane. So they are picking up on the vibrations in the fluid of that cochlear duct, right? So if we go back and look at our cochlear duct over here, that's this big area, okay, where the spiral organ sits. And so that's made of fluid or endolymph, and so that vibration of sound is going to be vibrating the fluid, which in turn vibrates these hair cells, okay? So these inner hair cells are picking up that vibration and transmitting it to the nerve. Now the outer hair cells just kind of fine-tune the cochlea. They can amplify the signal if needed. So these guys just have a very specific job, okay? But in general, just kind of know that these hair cells are what are picking up the vibration, the sound vibrations, and sending it via that basilar membrane into the cochlear nerve. Okay. So this picture just puts it all together, right? We start with that, with our bony labyrinth, and we take a cross section of the cochlea to kind of look at our different spirals, right? And then we blow up one of those spirals to look at those ducts, and we are most concerned with our cochlear duct because it contains that spiral organ. And the spiral organ is what houses those hair cells. Okay, so those hair cells are the main players, the receptor cells that pick up the vibrations and send it to the brain. So this picture just kind of gives you an idea of what the video actually shows. So you pick up this sound vibration coming in through the auditory canal, vibrating that tympanic membrane, which vibrates all those little bones, those auditory ossicles. And then that stapes, that last one, is going to vibrate that oval window, sending that vibration through these membranous ducts. Okay, and the cochlear duct is what we are worried about because it houses those hair cells, which will then turn that signal into a nerve impulse through that cochlear nerve and into the brain. So here's a link to that hearing video, and hopefully that'll help kind of explain what I just talked through on the last slide. I have posted the link on Canvas as well, so you can look at it there, and just hopefully that'll explain everything a little bit better. So the second inner ear structure we'll talk about is the vestibule. And we're talking about the vestibule, we're talking about the bony labyrinth, right? And it lies just medial to that middle ear with those ossicles. And within the vestibule, we had those two membranous structures called the utricle and the saccule. So the utricle and the saccule are part of that membranous labyrinth, and within the utricle and the saccule we have these structures called macula. So in each utricle and saccule, we have a macula. And within the macula, we actually have this sensory epithelial tissue. So just like our other senses, we always have some sort of sensory epithelial tissue that is connected to our cranial nerve and our nervous system. So let's take a look at this macula structure and how it talks to the central nervous system. So if we look at the macula, so these guys are going to contain those receptor cells, which essentially are going to talk to the vestibular nerve. So remember, our cranial nerve 8 is both the vestibulocochlear nerve. So we split it up into the cochlear nerve going for hearing and the vestibular nerve going for balance and equilibrium. So how does that do that? So if we blow up one of these macula, we look and we see that we've got a bunch of epithelial cells here, and some of them are connected to the vestibular nerve fibers. Okay, and again, these cells that are connected to the nerve fibers are called hair cells. So they're very similar, so we aren't very creative with calling them a different name, right? We have hair cells within the cochlea, and now we have hair cells within the vestibule in these utricle and saccules. Okay, so these hair cells sit in amongst these other epithelial cells, and they have these stereocilia sitting up in this otolith membrane. So this otolith membrane contains these crystals, these calcium carbonate crystals called otoliths. And I think I was talking about it in lecture before somebody asked the question, and we have these little stones essentially inside our inner ear. So they tell us the movement of our head. So is the head at an upright position, or are we tilting our head? Because what happens is, with the force of gravity, those little stones are going to move forward and tilt forward. So they are then going to in turn tilt those little hair cells, which will then stimulate that vestibular nerve saying, hey, we've tilted our head forward, right, or backwards. So it allows us to sense the tilt of our head, which helps us with balance and equilibrium. So our last structure in our inner ear are the semicircular canals. So the semicircular canals are the bony structure, and within those semicircular canals we have the semicircular ducts. Okay, so the semicircular ducts are what contain the fluid within those canals. Now we have a couple different types of semicircular canals, okay? So we have an anterior and a posterior semicircular canal, and essentially they are what are going to tell us about the vertical plane, okay? So the anterior and the posterior, okay? So these are these two guys over here. So they're kind of up and down, so they kind of are in the vertical plane, right? And then our last one, our lateral semicircular canal, is in kind of the horizontal plane. So it's going to go opposite those other two. And that'll make sense why we need some in the vertical plane and why we need some in the horizontal plane in a minute, okay? So let's look at those up close. So now if we look within the semicircular canals, you have the semicircular ducts, the membranous part, and at the very end of those canals and ducts, you see this kind of enlarged area, and that's the membranous ampulla. So it's kind of this enlargement section towards the vestibule, and these guys house a very important structure. So within each of these ampullas, you get these structures called the crista ampullaris. And just like these other structures we've talked about, the macula out in the vestibule, the spiral organ in the cochlea, these guys house that sensory epithelium, okay? So these crista ampullaris, these little purple structures here within the ampulla are what are picking up on this sensory epithelium. So if we blow up one of those ampullas and look at the crista ampullaris, we can see that they have, surprise, surprise, receptor hair cells. So again, these epithelial cells have these very long hair fibers coming up off of them, okay? So these guys are important for rotational movement. So we said that those little otoliths, those stones in the macula, sense the tilt of our head. This is sensing our rotational acceleration or rotational movement. Because remember, we have fluid within these semicircular duct, so when we move our head in a rotational spin, it's going to move these hair cells, which are connected to fibers of the vestibular nerve as well. So let's take a look at that. So when we do a rotational movement, I'm sure all of us are going skating and doing these amazing turns, but say you have some sort of rotational acceleration. That fluid inside that semicircular duct is going to bend those hair cells, essentially whichever direction you decide to go, and it's going to signal those fibers in that vestibular nerve and tell your brain that you are doing this very complex movement, okay? So now we know how these receptor cells pick up the information, whether that's sound vibration or movement, right? Tilting movement, rotational movement, having to do with equilibrium and balance. The vestibular nerve and cochlear nerve are going to combine into that vestibulocochlear nerve, and then they're going to transmit that information, hopefully up to the cerebral cortex. And so just like we've talked about our other neural pathways, it's very similar, right? So this is just showing the auditory pathway. So we're picking up the information from those hair cells in the cochlea. We're going to exit to through that cochlear nerve and essentially up eventually all the way up to our primary auditory cortex, right? So of course we're going to go through parts of the brainstem that you don't need to really worry about, but again, our big player is the thalamus, right? So all that sensory information is going to eventually pass through the thalamus on its way and send it to that primary auditory cortex in the temporal lobe, which makes sense, right? All those structures are in the temporal bone, and so we have the temporal lobe as well. Okay? So the difference of the pathway for the equilibrium pathway compared to the auditory pathway, eventually everything is going to get to the cerebral cortex. But the big thing with the equilibrium pathway that I want you to remember is it actually bypasses and it stops at the lower brain centers. So in the brain stem, you essentially have these reflex centers. So if you ever notice, if you are falling or have a balance problem, you usually catch yourself before you fall without even realizing that you are catching yourself, right? So this reflex center allows your body to react a lot sooner than if you had to send the information all the way up to the cortex and all the way back or to the cerebellum. So essentially it bypasses the cerebral cortex and has this reflexive response so that you can catch yourself before you fall. So obviously it does send the information to the cerebellum and the cerebral cortex, but those are really for those more complex movements. If you were to have a fall, it has a reflexive response, which is kind of interesting and it makes sense. So there are some disorders with the hearing and equilibrium, right? We know about deafness, but a lot of people get motion sickness as well. And they don't know exactly what causes motion sickness, but the popular theory is that there's really this mismatch of information. Your visual sense is not matching your equilibrium sense, and that mismatch of information is what can cause the motion sickness. Now, deafness is really any loss of hearing, right? But there can be two kind of main causes of deafness. Either you can have a conduction deafness where the information just cannot get to the inner ear. Those sound vibrations are being disrupted. Whether your tympanic membrane is ruptured, you have an ear infection so that middle ear or inner ear is full of fluid, that information just can't get to the inner ear and to the nerve. Maybe the ossicles have a problem. Versus a sensory neural deafness has a problem with that auditory pathway. So it can be somewhere in the central nervous system or in that cranial nerve itself. So it has something to do with the neural pathway, the auditory pathway, and not necessarily the conduction of the sound vibration. So these are the learning objectives for this lecture, and thank you for listening.